can get started, right? Everybody, I don't need to stand up. This is the session 2 of our microscopy at large sessions, and we're very happy to have here today Dr. Matt Brown from the Department of Biological Sciences at the LSU main campus, and Dr. Wu. Both of them direct core facilities microscopy here at the main campus as well as the vet school, Dr. Wu. And they will talk to us about their capabilities, equipment, and applications. And I don't know who's going to start first. Matt, you're going to go first. Okay. Get your mic on.
Yeah.
Make sure it's green.
It's green.
Yeah.
Okay. I'm going to just go over here. And
now, these sessions are recorded, as you know, and they will be available to students, undergraduate, grad students, or faculty later on for their view at their own leisure.
Okay, I guess I'll start. My name is Matthew Brown. I'm from down in the basement, the Socolofsky Microscopy Center. And I'm going to talk, my talk's going to be pretty basic. I'm going to talk first about the capabilities of the SMC, and then I'm going to talk about one minor application in fluorescence that gives people problems from time to time. So I'll go ahead and start. So our facility is located in the basement of the Life Sciences building here on LSU's campus. It comprises about 5 total rooms, taking up the majority of the basement. So we used to have 3 staff people, but the recent budget cuts to LSU have ended one of our staff funding lines. And so now we're down to Ying Xiao and myself who are staffing the microscopy center. Ying is our EM specialist. She has over 17 years of experience with electron microscopy and, more importantly, biological sample preparation for TEM and SEM. Biological sample preparation is a little bit, everyone who's done EM work with biological samples knows that it's a little bit more difficult than for non-biological samples and takes a lot of care. She has a lot of work. She gets a lot of work for preparation and sectioning of especially transmission electron microscopy specimens. I've been the light microscopy specialist since January 2007. I have some experience in cryotomy and sample preparation for light microscopy, but my real expertise is just with the instruments and getting the very best possible quality data from your particular sample or specimen. Our facility has a professor who is the more or less faculty liaison between the needs of the facility and the higher political apparatus of the department, and that's Pat DeMario. He basically spearheads all our grants. So our microscopy center has a wide user base. It possibly has the widest user base of any microscopy center in this immediate area and possibly the state. We assisted, just last fiscal year, we assisted 75 different labs from 20 separate LSU departments. And we also had users, not counting the users, from ULM, Louisiana Tech, and Tulane. And very occasionally we get work from outside sources who aren't affiliated with an academic institution. The stuff from ULM Tech and Tulane is partially spillover work. There was either a problem with their instrument at the time, or they needed some other particular thing that we could provide that they, for some reason, couldn't do. To accommodate this wide user base, our facility houses instruments that are, frankly, versatile and easy to use. That's what we value more than anything. Specialized instruments that have an extra layer of complexity and a narrowed amount of application, even though they might be more powerful and might be fancier, aren't of use to our facility really, because of we try to appease everyone. And ease of use, when we purchase new instruments, ease of use is very important. Pardon me 1 second. I think I know what's causing the problem.
Anyone with a BlackBerry will interfere with the wireless link.
Yeah, and I think my phone is close to it as well. sorry about that. Okay, now it's just, it'll be okay. Ease of use is very important because we let undergraduates and graduate students, we train them and let them use the instruments on their own after a suitable amount of training. So if an instrument is too complex to use or has a particular kind of catch that would make it fragile to people who are unfamiliar with these kind of instruments, it's not a very good instrument for our facility. All right, most exciting thing in our facility recently, as far as equipment-wise, is just at the beginning of this year, we got a brand new scanning electron microscope. It's a JEOL ISM-6610LV, and has should be, I want to point out, you see a red circle, we bought one specifically with a very large sample chamber. So that we can accommodate people who bring in rocks or metallurgical samples or material sciences things, so we can see that as well as our standard sputter-coated biological samples. Now, our transitional electron microscope is an older JEOL JEM 100CX-DEM. It's got a 100 kilovolt max accelerating voltage. We usually use it about 80 kilovolts. It's mostly an analog instrument. Basically, it's an analog instrument not because of choice, but because it's from the Reagan administration. But it turns out that this is easier to teach people on because analog instrumentation is more responsive than current digital instrumentation. The digital, to find the image and several other kind of parameters, as I've been told by Ying, are actually more difficult on the newer instruments because of the lag in response of digital controls versus the old analog. We use film to capture, again, because there wasn't really any available digital capture device when we purchased the instrument. But the benefit of film is it's much faster to capture the image. In fact, some people who buy new TEMs retain their old film capture or retain a film capture system because of this fact. And so this instrument, because of its age and the amount it's used, is the next instrument we plan on upgrading. So for my area, the instrument that probably gets the most use right now is our Leica TCS SP2 confocal. This is similar, I believe the vet school has one almost identical to it, and I'm pretty sure at least one other institution in the state has an SP2. It was a very popular instrument, and it's that for a reason. It's very versatile, fairly easy to use for a confocal, for a scanning laser confocal, and it had a decent price and good image quality, so it was very popular. So our TCS SP2 is a single photon system with a normal galvanometric scanner as opposed to a resonance scanner. Our confocal is equipped with 3 lasers that can generate 6 different excitation wavelengths listed here at 458, 476, 488, 514, 543, and 633. That hits most major commercial dyes or dyes that have been developed for fluorescence microscopy, but with 2 notable exceptions. Number 1 is DAPI. That's something that we constantly get requests for. And the second is a Texas Red, it's something around the 580, which maybe after we get the TEM, we'll be looking at upgrading the capabilities of our confocal, but that's a bit in the future. In place of important, if you don't have a Leica TCS SP2 at your facility, one of the most interesting things about it is that in place of emission filters, static emission filters, it uses a prism. It essentially sends the emitted light through a prism and uses mechanical baffles to only allow certain wavelengths of light to hit the detector. What this does is it gives you an adjustable emission filter. And this is a very important thing whenever you're trying to prevent bleed through between 2 samples. It's a great tool to assist in dye separation techniques. So another, like most every facility with light microscopy, we have just a general wide field compound microscope. So it's a DM RXA2 with this. We have a low noise CCD camera attached, and slide book. It's a software package called Slidebook. It's just an analytical software package attached to it. We have a wide range of filter cubes, 7 total filter cubes within this that cover everything from DAPI to Sci5 and everything in between, Texas Red, YFP, CFP, everything. Generally, if somebody brings me a fluorescent dye that fluoresces in the visible range, I can illuminate it using this microscope and the filter cube selection we have. We also have a wide variety of high quality lenses to attack various problems that people bring us, like immersion lenses, so that we can actually, and something that's fairly common, but not everyone has, a lens that can actually be submerged within like a growth media, so that you can see things within the growth media or on the bottom of a well. And yeah, it's a very, again, I've taken the thing apart and put it back together, it seems, over and over again, just because we have so many different kinds of needs of our user base for this microscope. An interesting microscope that we have is the Zeiss StereoLumar fluorescent stereo microscope. And so this is a fluorescent dissecting scope. So it's a low-powered fluorescence microscope. The usage pattern on this is weird. Not many people use it, but the people who do use it they use it a lot. And the reason is you get very unique images from this. Like the top left picture is, as I've labeled, a mouse retina infected with GFP labeled herpes simplex virus. And of course, you're not going to see the virus. You wouldn't see the virus with the compound scope. But you can actually see the infection pattern around the retina. And this makes a striking picture. Also, it's good for screening like large GFP expressing individuals. Like in this case, a Drosophila larvae with some have GFP, some don't. And you can basically weed out the ones pretty easily with this microscope. And another guy came in, and he had fed some insects he was interested in studying. He fed them rhodamine-laced food, and he was able to see their digestive pattern, see them take up the rhodamine food, and it lit up their digestive system, making a really neat picture. So our facility also has many other compound and stereo microscopes, as well as cameras, tripods, and light tables. We have a large array of instruments to prepare samples for light microscopy. Include, of course, sputter coaters, cryotomes, normal tomes for paraffin sectioning, ultramicrotomes for plastic sectioning, stains from a wide assortment of stains from a long time ago to things we just bought the other day. The real goal, as I see functionally of our facility, is for a user who has no background in microscopy can come to us and say, I want to see this thing in a certain format. In other words, they can bring us a sample and a journal that they want to replicate that kind of picture. And we should be able to do that for them. And we do a pretty good job of that. Now, I wanted to talk a little bit about a single type of fluorescence analysis, or fluorescence experiment, you could say, conducted with regularity in our facility. And that's colocalization analysis. Now, this is just one subset of fluorescence microscopy. And the reason I bring it up is it comes about once a month, a new user comes to me and wants to do some colocalization studies, which is fine. And this is one of it's a widely used application for fluorescence microscopy because it's very popular because it's very easy. When things have all been, when it's been properly controlled and you have experience with the various dyes. The purpose generally, as for biological samples, the purpose is to determine the subcellular location of a compound of interest. So now some things are going to be easy. So if you put in a compound or you have a fluorescently labeled protein, and let's say it goes to the nucleus, a colocalization study with DAPI or DRAC5, a nuclear DNA stain, it's not really that necessary. It's pretty obvious it goes to the giant nucleus there. But however, some other things like mitochondria or endosomes versus lysosomes versus Golgi bodies. That requires a little bit, they all essentially look like tiny dots within the cell. And so you have to establish by using usually commercially available dyes or a dye or a fluorescent protein that's already been determined to go to a certain organelle. You have to use that in conjunction with your protein of interest whose location you do not know. And so the good thing about this is for most applications and articles, a simple overlaid picture. You'll take an image of your sample and an image of the commercially available dye or the known dye that goes to a particular organelle, and overlay those. And hopefully those two, the two fluorescent entities that you put in there you'll either label them like a red and a green, or red and a blue, or a blue and a green, something when they overlay, they'll make a distinct color. Red and green is most popular because human beings can easily see yellow. So this is an image from a positive colocalization. This is somebody who had an unknown compound. They labeled their cells with mitotracker red. Their compound was green. They did an overlay. It's very easy to see a lot of yellow in the overlaid image. This of course is the raw green. This is the raw red, and this is the overlay. Now, for that kind of basic stuff, when it gets basic like that, it's actually pretty clear. But occasionally, you might need something more numeric to. You might need a more numeric representation to satisfy a reviewer or yourself that your two fluors are indeed colocalized. Now, a real popular representation to state colocalization is what's called the color scatter plot. And simply put, the color scatter plot shows the relationship between intensity, the intensity of various XY pixels for two different channels. In this case, the two channels are represented on two various axes. The red
it's a highlight. Green is highlight, sorry.
Oh, okay.
Blue or black.
Okay. Well, the blue will have to be the green.
Sorry.
In this case, the red is on the x-axis and the green's on the y-axis. And what this is really showing is the frequency of pixels at a certain intensity as their and their relationship with with pixels of the other channel. It gets a little confusing, but to make it easy to read, when you see a color scatter plot with the majority of the plot located along a 45-degree axis or in the center of the graph, that should indicate quickly that those two species are colocalized. Okay? Now, this is a different scenario where the top three are the red, green, and overlay channels. This is a certain kind of algae, and they've labeled something they're interested in with green, and I'm using a red channel to pick up the chlorophyll that's just natively around. And you can see through the overlaid image that the red and green don't really colocalize. And what you get here is a totally different plot. What you see is the red. I'm going to just stay right here. You can see on the color scatter plot, the red and green. They stay the dots on the plot stay closer to their respective axes, and this would indicate that the two channels don't colocalize. The two fluors don't colocalize. Now, again, I'm just briefly touching on this, and I'm just going to talk I'm almost done with my talk, but I want to point out a few practical considerations if you need to do this kind of experiment. The biggest thing that should be stated is probably that you need to bring controls. One of the big problems we have is, depending on the kind of dye you use, certain dyes bleed through to other channels. Now, with modern, if we're aware we're doing colocalization analysis and that channel separation is going to be a priority, we can make you can make provisions for that and how you set up your instrument. But it's still vitally important you bring negative controls to be, once you get a good colocalized image, you need negative controls with those same concentrations of dye and whatever kind of agent you're looking at, so that you can assure yourself that you're not seeing any bleed through with your colocalization. That's one of the good control set is vital. And without these, basically, you shouldn't even do it. You shouldn't even, don't bring your samples to the microscope. If you're going to look at colocalization, you have to have these controls with you. The other thing is, just for safety, you need to bring a sample, some of your sample that hasn't been labeled at all, just to see if there's any kind of autofluorescence in your sample. That will be just in case everything goes, if everything has gone wrong, at least that's the explanation, so you can know to go back to the drawing board. Now, if you haven't worked with a particular organelle stain, or if you're not really familiar with the fluorescent properties or the properties of the dye, if you're putting a dye or compound in your cell that you're looking at, you should probably look at the fluorescent reading. Basically, you should look at the image of your dye or your compound at different concentrations, and if it has a time, if there's a time component at different times as well, just to make sure that, well, as far as different concentrations, at low concentrations things appear not to be there. You don't get a signal. If you overload, if you put too much of certain kinds of dyes in cells, they actually start to show an erroneous localization, an artifactual localization phenotype. And so that destroys your experiment as well. The time point thing is that can be disastrous if your dye either takes a little bit of time to be absorbed within your sample, or if your dye is released from the sample after so many hours and if you've left it for a while. But those two things, it's important essentially for any scientific experiment. You should know what you're working with before you start comparing it to other things anyway. Now, as far as image analysis goes, the color scatter plots I showed you were generated using the Manders coefficient plugin available at, I used ImageJ. There's a great ImageJ plugin bundle called WCIF ImageJ bundle. If you have, it has lots of things in it, including this particular plugin I used to generate the colocalization color scatter plots. So you can download that in case your own, a lot of times microscopy imaging packages will actually have color scatter plot. You'll be able to generate those color scatter plots and other kind of data for you as well. Sometimes, depending on what kind of questions you're asking and where you intend to send your work to, you might have to generate more elaborate color scatter or colocalization information. There's a lot of that out there, and it's just beyond the time I have today to talk about that, and beyond my own knowledge actually, because most of the time all I have to do is do overlays and color scatter plots, and that seems to be sufficient for people. And that's all I have to say. Does anybody have any questions? All right, thanks.
So Matt,
yeah,
I can ask you a question. So typically, the corpus facility, there's a student coming up to you and they say, I want to do this, Matt, help me, and you tell them what to do. They bring you down there. So you're going to be assisting them through this on the first time around. You show them the ropes.
Oh yes.
How to do this, because obviously they can't get in there and use an instrument, right?
No. If you come in my facility and I've never seen you before and you're using one of my instruments, I quiz you. I'm a big guy. I look like an ogre. And I start being very gruff and ask you why you're there, and generally, I don't have that kind of problem too much. I've had one or two people come in and think that they're just going to be cowboys and use my microscopes. But one of the things that I'm charged with doing is maintaining the microscopes. And uneducated users tend to create a lot of maintenance hazards for us. Other than that, yes, people come in to me a lot of times. I get a lot of people from chemistry recently who are trying to do work with biological samples. And so they come to me and have very little idea, actually, of what they're, how they're going to actually make this result happen. They might have a paper or something, and they want to see that figure with their samples. And I explain to them what they have to do, and then we hopefully have a good time.
You're available by email.
Absolutely.
I ask you, which set of dyes should I use to do colocalization or to get the best contrast?
If you're going to come to my facility, man, that makes me happy when you do these kind of things.
Exactly.
It makes it a lot easier for me. But yes, I'm available, yes, by email or by phone, either way, or drop by. All right, so I guess.
Any questions at ULM? Any questions for Matt?
No, we're good.
How about Louisiana Tech? You guys have any questions?
What's your email address?
mbrown7 at lsu.edu. So M as in Matthew, Brown, like my last name, number 7. And if you have any questions, even if you're not going to use our facility, if you have any questions about something you're going to do and you just want to see if I have any experience with that, I'm glad to help. So,
Thanks, man.
hey, no problem.
Thank you.